Yes, I think we will start the
Okay.
session. Sensei, can
Ya,
we start the seminar?
ya.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Halo Pak.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam.
Good morning. Good morning, ladies and gentlemen, esteemed college and distinguished guests, the Honorable Head of Research Center for Mining Technology, Dr. Anggoro Tri Mursito, the Honorable Professor Masayuki Sakakibara, and the Honorable Older Researcher of Research Center for Mining Technology BRIN. In this morning, it is my pleasure to welcome you to what promises to be an insightful and engaging seminar on phytoremediation of toxic heavy metal contaminated water and soil by aquatic microphyte Elecaris acicularis. My name is Hendra Prasetia, and I will be your moderator for today's seminar. The seminar today will delve into the fascinating world of phytoremediation technique application to the radionuclear contamination in Japan. Professor
Ini orang
Sakakibara
lingkungan ini
is
judulnya.
one of reputable researcher in Japan to remediate cesium contamination and has a patent about the application. Before we continue to our special seminar, let us listen the opening remarks that will be delivered by Head of Research Center for Mining Technology. Please welcome Dr. Anggoro Tri Mursito. Anggoro, time is yours.
Thank you, Pak Hendra. Good morning, everyone. I think this is a special time for us and also to able to hold an online Ngopi Minerba between Japan and Indonesia. And also many thanks for all of us for taking the times to join and also attend this public lecture at the Center for Mining Technology BRIN. As Pak Hendra mentioned previously, that Sakakibara Sensei is one of the expertise in environmental pollutions And also he conduct many intensive work, I think, in the field work and also activity, especially for reducing of mercury pollutions and poverty problems in ASGM, yeah, in artisanal and also small scale gold mining. And Sakakibara Sensei is
Yeah.
is a professor which has a multidisciplinary background, I think, right, Sensei? You did a lot of work and also study, especially in geology, geochemistry, right? Geomedic, geoengineering, and also remediation engineering. Sorry if some field that I mentioned is left behind. So and as we know that phytoremediation is one of the technology that use plants to remove any pollution, any pollutant, sorry, from the environment and should be what is called like to be economic and also environmentally friendly and also energy efficient, because phytoremediation can be applied especially in a large area, and it is also useful for treating of a wide variety of contaminants, especially for metals. And in artisanal small scale gold mining in Indonesia, there are a lot of problems, I think, related to this kind of environmental what is called pollutions. And Sakakibara Sensei also has a quite experience in a field or in a laboratory work, and also he done a lot of research, applied research work in Indonesia, especially in Sulawesi, right, Sensei, and in Lampung, as well as in special region in Asia, something like that. And I think it won't be take too long for us to listen and also to discuss with Sakakibara Sensei, and hope to get a new insight, especially in the field of mining remediation. Thank you, Sensei, and time is yours.
Okay.
Assalamualaikum warahmatullahi wabarakatuh.
Okay.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for Dr. Anggoro Tri Mursito as head of Research Center for Mining Technology for the welcoming speech. For your information, the end of the session will be followed by short and question and answer segment. So please feel free to jot down your questions and engage with our speakers during those times. Before we dive into the seminar, let me briefly introduce you about the Professor Sakakibara research experience. Sensei, let me stop your share screen, and I want to share your curriculum vitae for a moment.
Moderatornya siapa?
Okay. Today, our special guest, he is Professor Masayuki Sakakibara, PhD. Masayuki Sakakibara is an earth scientist with a multidisciplinary background, such as geology, petrology, astrobiology, geochemistry, medical geology, geoengineering, and remediation engineering. He was born in Hokkaido, October 30, 1959. And current position is a special aide to the President of Faculty of Collaborative Regional Innovation, Graduate School of Science and Engineering, Ehime University. And he was also director of SWIGI, Six University Initiative Japan Indonesia. Then he was director of Asia Africa Center, Institute for International Relations Center for Disaster Management Information Research. And these are some contact address if you the audience would like to contact directly to the Professor Sakakibara Sensei. For academic qualification, he was graduated from Hokkaido University, starting from undergraduate until doctor course. All of the academic qualification was graduated from Hokkaido University. Then the working experience starting from 1988, he is a research fellow of the Japanese Society for Promotion of Science. Then become the professor of Department of Earth Sciences, Faculty of Science at Ehime University since 2005. And now he is a professor of BRIN, then also visiting professor of BRIN. BRIN is one of the research center in Japan. BRIN is research Institute for Humanity and Nature, yeah. And these are major publication that are related to our seminar today. Probably maybe you can share by yourself, yeah. Okay, that's all about the curriculum vitae of Masayuki Sakakibara Sensei. And then now, without further ado, let's begin our session. Professor Sakakibara Sensei, the time is yours. Please, Sensei, can you share your presentation file?
これはどうしたらいいんだろうな。これか。これでいいのかな。これか。これか。Can you see?
You're on progress.
So,
can you change into audience view? Sensei?
OK。これまた大谷さんさっきのやつどうしたらいいんだろう。オーディエンスのやつと変えるというのはどうしたらいいんだっけ。これが見えてるみたいなんだけど、今は。こっちじゃなくて、これが見えてるみたいな。それで、どれいや、違う。一回これ閉じてさ、俺の紹介してたから、それで今どうしたらいいのかがちょっとわかんないんだけど。これかなこれじゃないよね。これデュアルスクリーンになってるんだよね、多分。で、こっちをスクリーンにしたいんだけどさ、見えてるの。これをだから共有のやつを、これを選べばいいのかな。ダメだね。
Great, Sensei.
It's okay?
Okay.
Okay. I see. Okay.
You may start it.
Okay.
Thank you very much.
Good morning, everyone. So my name is Masayuki Sakakibara. I'm a professor in Ehime University, Japan. Today, I will talk about phytoremediation, so just experimental work in Japan. This plant name is aquatic macrophyte, the Elocaris scuraris. This one is very useful, also for Indonesia. And then after my presentation, shall we discuss for your future work? Okay. Okay. Commonly, so just phytoremediation, what is the phytoremediation? And then phytoremediation technology remediates the pollution of water, pollutant of water, and then soil, commonly water and soils by harmful heavy metals. Commonly, we use the physical-chemical technology and the phytoremediation. Of course, physical-chemical technologies are much better than phytoremediation because the phytoremediation need a time, and then we have to choose the plant. But this technology is very useful for economical, so just condition, and then also environmental friendly. The phytoremediation is a technology using just living plants or in situ or in place. The removal, degradation, or contaminant contaminant in soil and sludge and sediment and then surface water and then groundwater. So first, I want to explain the hyperaccumulator. Electrocalcium scale is also hyperaccumulator. The hyperaccumulator means just a plant that can accumulate large quantity of trace element mainly heavy metals from its environment. This is a category of heavy hyperaccumulator. The mercury is more than 10 milligram per kilogram, cadmium 100 milligram per kilogram, and the copper, arsenic, lead, nickel, cobalt, chromium, 1000 milligram per kilogram, and the manganese, zinc, more than 10,000 milligram per kilogram. This is the hyperaccumulators levels, and then hyperaccumulator is generally is a plant that can accumulate large quantity of trace element from its environment. So today, I want to introduce the Electrocalcium scuralis. This is belong to the Cyprosses. Cyprosses is a family of just a monocot. Commonly, this is a tiny, you can see the tiny flower. This is a scale. I didn't show that scale, but the common height is around 5 to 10 centimeters. And then this is very small with small flowers on land. This is aquatic, commonly aquatic plants. And then just very strong fertility that propagate mainly by root. Of course, they have a seed, but mainly by root. This means just not so easy expand. Of course, human beings can control the propagation. This is already we study on this one. So on the hyperaccumulator, arsenic, zinc, copper, and cadmium, and then lead. Also more in the laboratory. Of course, we just study the laboratory work, and then just more than maybe 20 elements. But maybe if you study on this one, just almost heavy metals accumulate by this plant. But we don't know the mechanism. So by accumulation, if somebody wants to study that one, it's a very important problem. So, and also being worldwide distributed. This means that in Indonesia also native plants. This is also very important for Indonesian, for BRIN researchers. Okay, today I want to introduce a two field experiment by this plant. One is the experiment, this is the we call field experiment 1. This is the heavy metal, polluted water and soils, phytoremediation in Okayama Prefecture, Japan. Okayama in the Western Japan. And then second one is a field experiment 2. This one is radiogenic cesium, so polluted soil in Fukushima. Maybe you remember the Fukushima nuclear power plant accident. This accident, so just radioactive, so just cesium pollution. And then so just I want to introduce these two experiments. Okay, and then the first experiment, so just the goals to develop and then apply the phytoremediation of toxic heavy metal contaminated water by using the aquatic macrophyte Elocaris ascularis. Okay, and then so just a materials method. The sampling the water, soil, and the plant. We do the sampling for the water, soil, and the plant for analysis. Okay, and then before, and then also collected before and then after transplanting. The analytical method is on the soil samples by XRF. This is Phillips. And then also plant tissue by Pixie. This is the Cyclotron Research Center of just Iwate Medical University. Iwate is northeastern Japan. This medical just university have this Pixie, and then so we are not use this one. But now this Pixie instrument commonly generally so just closed before maybe five years ago, around 5 years ago. And then now so Kyoto University can use now. Okay, and then also we use the ICP mass. This is parking error mass in Ehime University. But unfortunately, so Ehime University, this ICP mass also closed. Now we don't have ICP mass. This is the field experiment 1, so just experimental, so just map, land map. So this is the pond. We use the pond before dam. Okay, you can understand. The blue so just portion is a pond and then river. Okay, and then before dam. So this is this photograph here. Okay, this is the dam waters. And then so just we cultivate the Elocaris ascularis by using 3 methods. Okay, one is the floating cultivation, this one, and another one is the pot cultivation, this yellow one, and then so third one is the direct cultivation. Direct means on the polluted soils. This we use the 3 methods, and then we so just start the experiment. This is a direct cultivation. This is we transplanted directly on the polluted soils here and here also. But this cultivation method is probable because then so just that transplanted Elocaris ascularis flow out after heavy rains. This is a very important phenomenon. And then phytoremediation, if you start the phytoremediation experiment, you have to think about natural water conditions, especially in Indonesia. So sometimes, so Indonesia have heavy rains, and then if you cultivate directly on the polluted soils, you have a same problem. Okay. And the second one is the pot cultivation. This is using so just a container, and then we cultivate it on the so just sheet using pot. But this method also maybe some of mud covered so just plants, and also maybe can get the effect on the pollutants in the soils. This is also not so good. Later I compare, but advantage is that decrease in toxic by heavy metals leach, because we put the soils and then soil clean and then only water pollutants. So just this plant can absorb, but sometimes water level become high, and then so just some of mud touch on the Elocaris ascaris, and then sometimes they have a problem. Okay. And then next, third, floating cultivation. This one is we use the container also, but we put the PET bottle, vacant PET bottle, and then so just we float this container. Okay, and this method is very cheap and also very easy that we can use this one. And then advantage is the remediation of polluted waters by hydroponic cultures. Okay, and then measure to the rapid change of water levels by heavy rains. Because the water level become high, this so just the container also float, and then so on the surface by PET bottles. They control this PET bottle control the water levels. Okay, and then so just we show the so just photographs. This is the pot cultivation method. This method also not so bad, but sometimes so just cover some mud on the surface. Okay, and then sometimes have a problem on the growth, so just plant volumes. This is the biomass favorably so just increase, but clay material covered the shoot after heavy rains. Okay, and then this one is the floating cultivation. This one is the floating cultivation from April to October, or same so just a container, and then also dry weight of shoot and the root. We measure the dry weight, and then finally increase the so shoot around 2.5 times, and then this is very good for the cultivation because I'm floating on the pond surface, and then so cultivate well, grow well, and then so just finally so just we take these samples. This method can be applied to the change of water levels, and the biomass remarkably increase. Okay, and then so just I compared the three methods, and then so floating cultivation is the best method on the pond, or maybe river also. Okay, and then so just then next we do the so just feasibility experimental study, and then this one is on the pond. We put the so just container and the connecting everything, and then connecting this pond, connecting floating on this pond, and then we cultivate it. Okay, and then so every month we analyze the so just the water heavy metal concentration in of the water. Sorry. Okay. This is the data. So this data is so very interesting one. Okay, first, yellow one is red one is so just upstream, and then yellow one is this is the river water upstream, and then yellow one is lower stream, and then green one is the pond waters. And then, so just the blue line is the boundary of, so just before cultivation, before experiment, and then after experiment, this is the boundary, okay? And then we can compare, we can watch the, so just how to change the heavy metal concentration. This is the manganese, copper, zinc, arsenic, cadmium, the lead. And then so just each water levels, each concentration levels, we check. Okay, and then so we just after cultivations, arsenic here, arsenic, and then copper here. The concentration levels become low after cultivation. Example, so just on the arsenic, okay, upstream levels almost the same, but okay, after stream, before stream, before pond, that one almost same, but after pond, in the after dam, so concentration levels will become low, and then also pond level also become low on the arsenic. Okay, and the lead also almost same. This has been very useful for arsenic and lead. This is for the feasibility study experiment work. Also, very important one is so just material balance. We check the material balance. Maybe you know the material balance. So material balance is how to check that one. I want to show the process. The first, we estimate the boundary of polluted river waters in 1 day. This is for the depth less than 0.005 meter per second, and then 24 hours, and then 60 minutes, and then 60 seconds. This is the totally, so just 1 day, so just a water pillar, okay? And then so just we can calculate the arsenic. We already checked the arsenic levels, and then okay, then so just oh, sorry. Okay. Okay. And then arsenic concentration around 50 microgram per liter. Okay, this river water. So before pond, and then total arsenic weight of river water per day, 5.4 grams of arsenic. Okay, this is we estimate this one, and then how much Eleocalcis accumulate this one. So we also calculate this one. So Eleocalcis accumulates just arsenic per day around 100 milligram per kilogram. Okay, this is already my colleague just study this one, and then we calculate this one accumulation weight of arsenic by Eleocalcis per day. this Edocarditis is around 360 kilograms, and then totally 3.6 grams arsenic, 3.6 grams. This means that around more than 60% absorb this 360 grams. So just the Edocarditis scuraris absorbs so 3.6 grams arsenic per day. This means so just this phytoremediation, so just theoretically, so just it's possible. And then also maybe our data in the pond, so this is also maybe consistently we can get the result. Okay, and then so just I move to the next experiment. So this plant also can absorb the cesium, and then so just we try to so just remediate so just radiogenic cesium pollution. Do you remember the maybe? Do you remember 2011's Tohoku earthquake in Japan? It's a very famous. So this green this so area so just tsunami attack around here, and then also many people die. This tsunami height is become 30 meters, maximum around 40 meters. Okay, but this one is a tsunami photograph. And then also maybe at the time, so just the earthquake and the flooding, so just attack the nuclear power plants in Fukushima. This is the explosion, hydrogen explosion. And then so just this dust flowed by the window and then covered by the large area. Okay. And then so just this is the polluted areas by the radiogenic cesiums. This is already so just measured by the mixed and then DOE. Radiogenic cesium, so just from this is the nuclear power plant here, and then so move to the by window, move to here, and then go to southwest, and the wider area around maybe 200 kilometers. Very large areas are polluted by radiogenic cesium. And then we try to so remediate this one. So, and then so first we do the 2x. Okay, one is the native Edocarditis scuraris. We measured so just 134 cesium and then 137 cesium of native so Edocarditis scuraris in Fukushima prefectures. And the second one is we transplanted so Edocarditis scuraris in the soil of so paddy field Fukushima prefectures. We do these two experiments and then cultivate, and then so after experiment we measured 134 cesium and 137 cesium. So this one is also maybe using so after sampling, so we measure by the this one, so germanium, so just detectors. This one is very useful for us. And then, so I want to show the result. This one is the native, so just Edocarditis scuraris after accident in the paddy field. Already so grow up. This one is native electrolytic saccharides. Okay, and then this one is in Koriyama City, Fukushima Prefecture. So, and then this one is also another area. So of course, so just a farmer didn't cultivate the rice in this year, 2012, but electrolytic cultivate grow up well. So instead of rice, and then we took this sample and then checked the just radiogenic cesium. This is why the data. Koriyama FATC, the agriculture, so just a research center in Koriyama, and then Kuzuo, and then Namie town. So this is blue one is the soil, and then so red one is the electrolytic saccharides. This one is very high, around 6,000, more than 6,000 becquerel per kilogram. Okay, but Namie is very low. How to understand this differences? This means that so just commonly also in natural soils. Of course, non-radiogenic cesium also remain in the soils. And then if the soils includes non-radiogenic cesium, so maybe totally, so just radiogenic cesium so become low, because then so non-radiogenic cesium includes non-radiogenic cesium. We didn't check the analysis, but maybe I think if electrolytic saccharides ability are same in everywhere, so this difference so just between the so just non-radiogenic cesium content of soils. Okay, and then so we cultivate the electrolytic saccharides artificially, so just in the paddy field. This one cultivated August 11th. Okay, that why so after cultivation, so September 4, after 3 weeks, we took the samples on the same place. Okay, and then this one is another experiment, also cultivate the electrolytic in the paddy field. Okay, and then this one is so after cultivation, the electrolytic saccharides grow well. Okay, and then so just another experiment also, maybe we put so just electrolytic saccharides in the paddy field and then grow well like this. And then we took the samples. That one is the result. One is the soils, so just a becquerel, different becquerels. This is the 15,400 becquerel per kilogram, very high, and then 3,800 becquerel per kilogram. And the blue one is so just this is a different same place and the different years. But after 2 months, the maximum so just the becquerel so per kilograms in each experiment become higher. But the blue one is only one time, but the red one is maybe gradually high, but some of them become low. But the maximum level is almost straight line, become straight lines. This means that so just electrolytic saccharides absorb the cesium. Even so, just potassium, so just even potassium, so just this means the electrolytic saccharides absorb favorably, so just from the paddy soils. And this one is also just another data. So just the Japanese government tried to cultivate just sunflowers, because on Chernobyl, so just accident, after Chernobyl accident, so just the scientists cultivate the sunflower and then can absorb the radiogenic cesium, and then Japanese government also tried to do, but radiogenic cesium, these sunflower couldn't so just absorb, because then so just maybe this is my opinion. So just if potassium rich and not it's potassium rich, commonly sunflower so can favorably so just absorb the potassium compare with so just cesium, and then potassium rich soils. In the potassium rich soil, commonly sunflower can favorably so just absorb the so just potassium, and then so just this means so just sunflowers, so these sunflower couldn't so absorb the radiogenic cesium, only 0.0128%. Okay, this one is also very important. And then this paddy field, commonly the farmer puts a lot of so potassium, and then maybe sunflower we couldn't use the sunflower. But this one is the Chernobyl area, because then so sunflower camps, not only sunflower, amaranthus also can absorb. Because then these soils maybe they didn't so inform us on the potassium content, but maybe this is the continental areas, potassium not so much. And then so just they can absorb this radiogenic cesium. Maybe this result may be showing the so just the radiogenic cesium concentration, so just with the potassium. It's very important when we do the so just radiogenic cesium pollution remediation. So just we have to check the potassium content, and then also non-radiogenic so cesium concentration. Okay, this is the final slide. The potential of the electrolysis to take up the radiogenic cesium has been established by the screening the plant, and then so just cultivate the experiment. Our experimental data elucidated so that electrolysis ascularis is the most suitable for phytoremediation of radiocesium, so just from contaminated soils. But the most important portion is so just non-radiogenic cesium content. We have to check that one. So when we do the so just phytoremediation work by electrolysis ascularis in the radiogenic cesium pollution area. Okay. Thank you very much for your attention.
Thank you very much, Sensei,
Yeah.
for
Okay.
very nice presentation.
Okay.
This is very insightful for us
Mm.
about the electrolysis agriculture.
Mm.
Before we move to question and answer session, we have a photo session.
Mm.
Photo session first. Please turn
Okay.
on your camera. Bapak
Okay.
Ibu dipersilakan untuk mengaktifkan fitur kameranya. Kita ada sesi foto bersama terlebih dahulu.
Mm.
Okay, we have a photo session. Maybe Bu Lisa, you should take the picture. Okay. We have three slides here.
Okay.
First slide, 1, 2, 3. Okay, Lisa. Then second slide, 1, 2, 3. Okay. Next, last one. 1, 2, 3. Okay, Bu Lisa. Dan sudah, Bu Lisa. Okay. Thank you very much, Bu Lisa. Okay. Now we dive into question and answer session. If anybody would like to ask Sakakibara Sensei, please using feature raise hand in the Zoom or any chat about the presentation. Okay, Sensei. Maybe starting from me, I would like to ask about the Elocarus acicularis.
Okay.
Is there any similar type of Elocarus acicularis plant in Indonesia? Have you ever
Yes,
found?
I already bought maybe around 10 years ago in the aquarium shop.
Aquarium shop.
Yes, maybe I think a shop owner so just puts the name. This is the cow hair.
Okay.
This is maybe just a name, and the aquarium put into the aquarium. So just maybe aquariums in the aquarium they put into there for the fish. And then so just this means so just commonly so Indonesian can get so just Elocarus easily.
Oh, so
Yeah.
it is very similar phenotype genotype with the
Yeah,
Elocarus acicularis.
I didn't check it, but maybe I think so. Yeah.
Okay, it's
Yeah.
interesting because from your presentation, Elocarus acicularis could observe high concentration of arsenic, sulfur, and also cesium.
Yeah.
Then, Sensei, my next question is, as we know that the phytoremediation technique avoid using edible plant, yeah, commonly not using edible plant, consumable plant. How about the Elocarus acicularis? Is it edible? Some animal can eat this plant or
No,
any
never.
yeah, because we avoid the food chain contamination. Yeah. If
Yes.
it is eaten by the animal.
Because I know almost this so shoot is this is monocot, and then also no almost this is the leaves, but this is the leaves, this tiny so leaves, but maybe not for tasty. I don't know, not tasty, and then for animal, and
I
then
see.
also maybe flower is also maybe no insect, so maybe touch on these flowers.
I see. So
Yeah.
it is very good. Yeah. For
Yes.
our
Yeah.
phytoremediation technique. Yeah. Okay.
And
Then
also maybe so lot of so just plants grow so just in the under the so just highly so just grow, and then also maybe not so easy not so easily so expand to other, because then commonly they propagated by the so just root,
right.
and then so just not so easy expand. Then we can control so just cultivation. This is also
Okay.
very useful.
And then my last question, Sensei.
Yeah.
This is a very important to know that after the Radicals acrylaris absorb the heavy metals
Yeah,
and radioactive cesium.
yeah, yeah.
How to storage how
Okay.
to storage the plant so we can
it's
keep the
yeah.
heavy metals inside the
Oh,
plant, not
yeah.
release to the
It's
environment again.
it's very important for phytoremediation. So just after phytoremediation, so in Japan commonly, so we so just we burned so just waste materials. So very serious one is maybe of course under the very severe conditions. So severe means so just using filter or something, and then
Okay.
never expand. But in Indonesia, maybe dry up. Okay, don't need burning, but dry up, and then so just bury. Because when you burn the ascularis, so absorb the heavy metals. So it means so just during burning, so just ash expand, so just other area, and then sometimes so just smoke includes heavy metals, and then so now so don't need burning. After so just taking the so just after taking electrical securities after experiment, so and then dry up, maybe after dry up, so just the volume become 10%. Okay, and then also maybe easily can make the storage. Yeah.
Okay.
But the burning is better because after burning, the volume become 1%. This means almost waters and then also light elements.
During the burning process, is there any filter?
Yeah, need so just
Special
filtering. Yeah, because
is
the
there
smoke, so just never expand the smoke.
any special material for the filters?
On the heavy metal, I think I don't need, but radiogenic, on the radiogenic cesium pollution, maybe we need a filter.
I see.
But maybe first step is so the Indonesian so just electrical scales can absorb the so just heavy metal or not. Nobody knows that one.
Okay.
Yeah, that one. If you can check the so just ability, can cultivate many places. And then so just after cultivation, so after grow up, so just you can take the sample and then dry up and then keep in the storages. But maybe finally, so just we have to do the burn.
Burn. Okay.
Okay. Yeah.
That's the final
Yeah.
solution.
But
Is
if
burn.
somebody so have an interest on the in this electrical scales, where is the heavy metals? We already do some experiment. Electrical scales, so just this monocoat, so just have silicon.
Silicon.
Yes, plant opal inside. Okay.
Okay.
Yeah, it's a very useful one. So this plant is a opal, and then this mineral very stable. Maybe during so just more than 5,000 years, and then if these heavy metals so just can enter so just this so plant opal in this plant opal, maybe this means very stable, and then also plant opal so just absorb these heavy metal, so just after burning also heavy metal in the plant opal. if
I
we
see.
can check this one, so just maybe after burning, it's okay. Maybe put into the soil also, no problem. Because the plant opal is very stable. Why so just we can say that very stable? Because in Japan, commonly, Jomon era, Jomon is an example around 6,000 years ago. Plant opal, still we can detect, we can find the plant opal from the soil. You know? This means that so just plant opal very stable, and then also they shape the they keep their own shape.
Yeah.
Yeah, this means that never so just break up, and also never so just melt, or maybe, and then so plant opal can keep the so just their own so shape, and then this means that
Never
if
deform.
yes, and then so just we have to check the this ability.
Okay.
I check that only one sample, so just heavy metal so in the plant opal.
The plant opal located in the shoot part or in the flower? Where
No,
exactly?
shoot.
In the shoot.
Yeah.
You didn't found in the root area?
I didn't check. So, but in the shoot area, there are many plant opal. Yeah, and then after maybe decomposed by the acid, so we can check the so just plant opal easily.
How to check? Is I mean
By the electron microscope.
electron
Yeah.
scanning electron.
Yes.
I see.
Yeah.
Okay, very nice information, Sensei.
Yeah. It's if you can use that one, so just please do it.
Yeah, we can try. Okay.
Yeah.
Okay, we have a question from audience, Sensei.
Okay.
First, if we are planting Alocaris articularis in between paddy field to absorb a heavy metal, how condensed is it? Then, what is the percentage of heavy metals removed by Alocaris articularis?
Okay. Maybe depend on the so just concentration of heavy metals in medium, okay, in the soils. But around maybe example on the arsenic, around maybe more than 1,000 ppm. And then also maybe I didn't remember the so just exact so just data, but okay, I will show the slide. Okay. Okay. Around this is the no plant data. Okay. No plant data. Sorry. So I didn't put the plant, but maybe few hundred to the maybe few thousand ppm around. Okay, and of course higher than another plant. This is this is the hyperkilometers. Okay, this means that more than this is the hyperkilometer levels, and then this I didn't remember the so exact data, but sorry.大谷さん、これちょっと消えちゃった。Sorry. スライドが閉じちゃったの。Can you see this one?
Not yet, Sensei. Maybe I will stop share screen and then you reshare the
Okay,
file.
I see. But anyway, so just more than hyperaccumulator levels. So just this means the very high. Okay.
Okay, Sensei. Maybe
Mm.
we move to
Okay.
the next question. We
Okay.
have
Yeah.
next question. What is the percentage reduction in radiation levels due to the spread of cesium after remediation using electric articular span in both soil and groundwater or river?
Sorry.
Percentage reduction level of cesium.
That might maybe also maybe we have to check the material balance. Okay, because radiogenic cesium is distributed in the wider area, and then if we cultivate the localized scaries, maybe we have to check the material balance. Material balance is the total weight. So in some area, total weight of the radiogenic cesium, and then also how much localized can absorb, how much percentage.
Yeah.
Yeah.
It means
I didn't calculate that one.
Yeah, I see.
Yeah.
Okay.
Yeah.
And then there's an interesting question, Sensei. The punish look like a grass. Yeah.
Sorry.
So like looks like a grass. Do you know?
Okay.
Okay. And then most animal especially in Indonesia, the animal eating the grass, consume the grass. Yeah. How to separate, how to distinguish this is the Eleocharis avipularis and this is the grass, because there's a similar type looks like. Do you understand the question too?
Yeah, I don't know. So just a very resemble just a plant.
In Japan commonly live in the soil or in the water center?
Commonly in the river.
In the river.
Yeah, sandy, so just the bottom, so just the lot of just grow up, but of course not everywhere. Of
So
course,
so
sandy.
the plant not live in the field, yeah, but in the bottom of
Yeah,
river.
yeah, but the farmers told me, so in the paddy field also the farmers just can absorb, can see the educational skills, but
I see.
Japanese government also creates the let's say the poisoning, chemical, materials, so for the eloquence skills, because it is eloquence skills grow up well, so just in the paddy field, and then they want to reduce the eloquence skills, and then they put a lot of chemical, yeah, material, and then just reduce the eloquence skills in the Japanese paddy field. Yeah, and then just not natural one. So just for the especially for the ascarius, it's a very interesting. They don't understand the electrolysis effect because they can absorb the arsenic. But maybe farmer hates the electrolysis ascarius absorb the fertilizer.
Yeah.
Yeah, and then they hesitate that one and then take off.
I see.
Yeah.
Because this plant, this plant is very interesting because it can live in the soil and water. Yeah. Based on your research experience, which one is better absorbed, in the soil or in the water medium?
Soil, commonly soil. because
Soils.
from the root.
I see.
Yeah. And then, but of course, if you put the container like a floating, so just floating method at the time, so maybe so aerocrystal can absorb the so just absorb the heavy metal from waters. Okay, but commonly, so aerocrystal grow up so in the on the soils, and then at the time, so just aerocrystal is absorb the heavy metal from the root,
I see.
from the soils.
From the soil. Okay.
Yeah, we can choose
Okay.
which medium, so just
Oh yeah, which medium we
yeah,
want.
yeah,
Then we have next question, Sensei.
okay.
Is it because your research experience also including the SGM activity that high mercury contamination? Then the question is based on your experience, what one is the best accumulate for mercury metal? How much is the percentage of removal and how long to get the
I
to
didn't
get
show
the
just
percentage?
I didn't show the so just yeah, it's a very important question. So just on the SGM, so just we so okay. This one is so just the last month I also did so the presentations in the BRIN other groups.
Yeah. In teknologi bersih.
Yeah.
Clean technology, sorry.
Yes. This one is a very also maybe これ大谷さんどうしたらいいんだっけ？これ違うスライドを見せたいんだけど、共有せないかもね。OK、共有で、これでいい、これなんだけどさ、これはこれだよね。新しいやつ。これでもこれで出ないんだけど、これ。これをそうか。これね。これだね。Can you see this one?
Yes.
Okay.
Can you change to
This one is maybe
audience view, Sensei? Audience view.
yeah. Audience, OK。これどうやってチェンジするんだっけ、大谷さん。こっちが見えてるらしいよ、左側が。これのこれね。これ。Can you see this one?
Yes.
Okay.
You can see clearly.
We do the tank experiments already.
Okay.
So just put into the this one is for the silicon effect, and then but using the so just many heavy metals, okay, and then we calculate this one. Okay, this one is a concentration, so just copper, zinc, cadmium, so lead, indium, silver, gallium, thallium. Okay, I didn't check the so just a mercury check, but you can understand indium, so silver, indium and silver, then lead, also zinc, also. This one is going down. This is puts a lot of so just silicon. This one concentration so in the water become low largely. Do you understand? This one without silicon or a small amount of silicon. Silicon is very important for absorb. Yeah. And then so this one almost heavy metal can absorb. This means so just maybe mercury also same. Yeah. But maybe next maybe experiment. So just we have to do the feasibility study. We can use the just this plant for phytoremediation in the field. Also, not to put the experiment, so just in the field experiment, and then so just we have to check the material balance, and then so how much can absorb, and then so we have to estimate. maybe systematic work.
Yeah.
Yeah.
For the real application.
Yes,
Yeah.
yes, yes. It's very important.
Very important. Yeah,
Mm,
because
mm.
okay, Sensei.
Yeah.
Next question.
Okay.
How is the performance of electronics articulating
Mm.
affected by environmental factors such as pH, temperature, or salinity? Is there any significant influence?
It's a very important. So just, but we didn't check the so just ask example. So just maybe pH and then also other factor. Please do it. So just I didn't check.
Okay.
Just
We
water.
will
So,
try.
yeah, yeah.
Okay.
Example, heavy, the mining area, commonly the waste waters commonly very acid, and then also we have to check that one. But grow up, so just I already found the eloquence ascaris in the acid waters, and then so just maybe so eloquence ascaris can survive in the so maybe acid waters, but not so high. Yeah, common.
Very interesting. It is
Yeah,
very tough.
yeah, very tough.
Very tough one. Yeah.
Yeah, and then so just eloquence ascaris never choose the so just metal.
Yeah. And
Yeah,
then, is it cheap, Sensei, to grow up
yeah, cheap.
the part, and how long it can grow big?
Yeah, never become big because the height is
Mhm.
maybe maximum 15 centimeter, but commonly
15.
around 10 centimeter.
Okay. I
But
see.
like a mat, the plant mat, like it's nothing like grass in the grass
Yeah,
garden.
like a grass.
Yeah,
Yes.
yeah. And then also maybe we already so just use this one so in the some area in Japan. But maybe if cutting off after absorbs those heavy metals and then cut off the so just cut off the so shoot, okay, and then later so just ascaris grow up, and then so just we can so just cut many times, and then can reduce the heavy metal concentration in the soils. Yeah, but we didn't check that this experiment. So just and then, if you have an interest on that one, so just please do it.
Okay.
I can support that. Yeah, if you want to do.
Thank you very much.
Yeah,
Okay, it is very interesting.
yeah.
Okay. Is there anybody, Bapak Ibu, ada yang ingin bertanya lagi? Mungkin jika ingin bertanya dalam bahasa Indonesia, bisa kami bantu untuk pertanyaan ditranslatekan. Oh, this is the data yang Sensei.
Yes,
You
yes.
you
Plant
compare
data.
between root and shoot.
Yes,
Yeah.
yes, yes. Mm.
I see.
But we don't know the mechanism, so just on the electrical absorbing so heavy metals. Because so common plants, maybe they have a favorite so just element.
Yeah.
Yeah, but electrical ascaris can absorb almost everything, all heavy metal. And then also example potassium and then cesium, commonly plants so favorite so just potassium. But in the electrical ascaris, this plant so just can absorb cesium with so in the so just potassium rich body. This means the so just why this plant so choose the cesium. We don't know that's the reason. Yeah, this means the now so still so unsolved problems. Yeah. But anyway, so just please do from the first step, please find the native soil localized queries in Indonesia.
Jadi similar.
Yeah,
Okay.
yeah, and then so just there are many. We need many works.
Yeah.
Mm.
We will try later.
Yeah.
Okay,
Easy.
very nice information, and
Yeah.
this plant have a very good ability to absorb
Yeah.
multi elements of heavy metal. Yeah. Okay, maybe no more question and answer,
Okay.
Sensei.
Yeah.
Thank you very much
Yeah. Sama-sama.
for a very nice presentation for today, and then happy. I would like to say happy retirement. This year you enter your retirement.
Yeah.
And then maybe we can join collaboration after your retirement later. You can say.
After retirement, I also maybe can support so just Indonesian researchers.
Please, Sensei,
Yeah.
please come again to Indonesia.
No problem. So,
Okay.
Yeah.
Okay. Maybe that's all. Thank you very much. Terima kasih untuk Bapak Ibu yang telah menghadiri acara seminar Ngopi Minerba pada seri ke-30 hari ini. Saya selaku moderator ingin mengucapkan terima kasih banyak. Mohon maaf jika ada kesalahan, baik perkataan yang kurang berkenan. Saya akhiri. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Jangan lupa Bapak Ibu untuk mengisi daftar kehadiran. Terima kasih. Thank you very much, Sensei, for your time.
Okay.
You may leave.
Yeah.
See you next meeting. Okay. Thank you very much.
Yeah.
Sampai berjumpa di Ngopi Minerba seri berikutnya.
Terima kasih Mas Hendra, Mbak Lisa,
Bu
semuanya.
Widi, gimana kabar?
Sama-sama Bu
Izin
Widi,
leave
saya selalu di US-nya Bu, ditunggu pulang.
Insya Allah.
Ya.
Doakan bisa pulang.
Terima kasih Bapak dan Ibu.
Oke, izin live ya semuanya.
Ya, Bapak Ibu silakan. Ibu Lisa, saya juga izin live ya.
Ya,
Terima kasih.
silakan. Stop dulu.